Hi, and welcome to our class meeting for Monday, April the 6th. And so we're going to start out with where we left off. So we went through neuron physiology and action potentials and all that kind of stuff, which is primarily the same as what we have in muscle tissue. And so now we need to look at when we synapse with a muscle cell, when the neurotransmitters come across, how do we get rid of those neurotransmitters? So that's where we're going to start. And okay, and get rid of me. Okay, so all right, so when we let's just go back a thing, look at this picture. Okay, so here we are at the synapse, and the neurotransmitter has released its the neuron has released its neurotransmitters across the gap, bound to the chemically gated sodium channel. Sodium channels opened up, sodium rushes in, depolarization. Now the cell has to have the opportunity to repolarize. So first of all, the only way that we can get the chemically gated channels to close is by removing the neurotransmitter. So there's three ways that the neurotransmitter can be removed. Okay, so if we look at this first part, it says if the neurotransmitter is not removed from the postsynaptic neuron or postsynaptic receptors, it'll block reception of the additional messages. So there are three ways that we get rid of our neurotransmitter. So the first one is just break up the neurotransmitter in the first place. So if we go back here, that's what's happening to this. We are degrading the neurotransmitter, and that's the process that happens with acetylcholine. So when a neuron is stimulating a muscle cell, then the acetylcholine will be broken up by an enzyme on the postsynaptic membrane. So next to where the chemically gated sodium channels are, and in fact, let's do this. Okay, so we get right with, let's get orange. All right, so right here, in and around where the chemically gated sodium channels are, okay, so an orange will be a little protein enzyme called ACHE, or acetylcholinesterase. So the neurotransmitter will leave this spot, go into here, and then it'll get broken down, like we see in that picture. All right, now, the second thing that can happen is that we can take it back up again. So we'll talk about astrocytes in lab tomorrow afternoon, but so don't worry about that right now. Or the presynaptic terminal for storage or destruction. That's the fate of norepinephrine, dopamine, and serotonin. And on Wednesday, we will look specifically at how this all happens, but let me just go back here. So what's going to happen in the case of norepinephrine, dopamine, and serotonin is that those neurotransmitters will bind, and then they'll come back off of the receptor and go back into the synapse. And then in the synapse, or on the postsynaptic membrane, then there will be receptors here for dopamine, serotonin, and norepinephrine. Okay, so then that'll just go back into here, and it'll we'll recycle it, and it'll get put back into a synaptic vesicle and stored again until it's needed. Okay, so we reuptake the neurotransmitter in some cases, or it just can diffuse away. Whoops. Okay, so it may just be diffusion away from the synapse, that it's not taken up by anything per se, but just isn't in there and enough to open up the channels and make a difference. Okay, so now we want to go on to, and I'm going to come back to this picture right here, to things called EPSPs. Now, we already talked about this last week, in that EPSPs, or local graded depolarizations. And so what that means is, in fact, I'm going to go to my whiteboard and draw some stuff on my whiteboard. All right, so we talked about this last week. So here is my neuron. Okay, and then I have my dendrites where I'm going to get a signal. All right, and so then I'm going to get synapsed by another neuron. So another neuron's going to come in here. And I'm going to have an axon terminal there and an axon terminal here, and maybe one over here, and one over here, one out here. Okay, so various different places where other axons can synapse with my no nucleus in there, synapse with my neuron. Okay, so if these are EPSPs, so we're gonna make these. If it's an EPSP, this means excitatory postsynaptic potential. Okay, so excitatory postsynaptic potential. So what that means is that the signal that's received by the neuron right here from this neuron here is going to be a depolarizing one. So if the neurotransmitter comes across the gap here and it binds, and then I get enough neurotransmitter to open enough chemically gated sodium channels so that enough sodium came in that it took the membrane to +4, okay? And then this, if this is a 5. So remember we did these last week. And this is a 2. Okay, so that's 11, so let's do a +4 right here. Okay, so every one of those is an EPSP. So these are plus a number, 1 or more. Just do that. Okay, and they are all depolarizing because they're all excitatory. Okay, so we'll come back to the definition in a minute, but then let's do IPSPs while we're at it. So then we have things called IPSP. Okay, so now my other remaining neurons, maybe this is a hyperpolarizing signal. So instead of sodium rushing in, and that's another point, I'll go back and draw that in. Instead of sodium coming in, either potassium is coming out or chloride is going in. So this could be a -5. This could be, a -1. Sorry, this could be a -3. Okay, so now an EPSP is going to be a -1 or more. And it's going to be hyperpolarizing. All right. Okay, and so this is going to make it less likely to fire. So let's put that in here. Less likely to fire. In other words, make an action potential. So I'm not going to get an action potential as much. And then how do we get that? So either we're going to open potassium channels. So I'll have K out. So I'm even more negative on the inside than I was before. Or open chloride channels, which has a negative charge, and then chloride in. Either way, I'm getting it more negative on the inside. Yeah, we haven't had chloride channels to this point yet. They are in neurons though. Okay, so let me go back and make this green and add one more thing. Okay, so this depolarizing, more likely to fire. Okay, and then we just open sodium. That's all we're gonna do here is open sodium gates. Let's see, what else? So more likely to generate an action potential. And they have various values. So you can see some +2, some +4, +5, minus +4, -3, -5, -1. So with EPSPs or IPSPs, they have varying amounts. Okay, so it doesn't matter. It just matters, is it a plus or is it a minus? And then, what was the other thing I was going to say? Let's take a look at the effects on this neuron right here. Okay, so if I go, what do I have? So I have 9, 13, 14, 15. So I have a +15. Okay, but then I have a, whoops. And then I have 5 and 3 is 8 and 9. I have a -9. So what's going to be my resting membrane potential altogether? Well, it's going to be plus 64. No, it's not plus. Oops. Undo. It's going to be minus 64. Because I took it, so the +15, I was at -70. I went to -55 with my EPSPs. But then I took it back down to -64 with my IPSPs. Okay, so that's how both of the graded potentials work. So let's go back to our notes. Let's see, how do I save? Okay, so I'll show that later. All right. All right, share. Okay, so here we are back in our EPSPs. So locally graded depolarization events at excitatory postsynaptic membranes. Each EPSP lasts for a few milliseconds in order to help trigger the axon potential at the axon hillock. If the currents reaching the hillock are strong enough, okay, so if those pluses we all added up stayed +15, then they would be enough to depolarize to threshold. Then the voltage gated channels would open up and the action potential is generated. So let's go back and look at why don't they last? Why don't these little depolarizing signals, why might they not add up to a threshold stimulus? Because here's what happens in dendrites and cell bodies, okay? They only have chemically gated sodium channels. So without voltage gated channels, as on a dendrite, voltage decays because the current leaks across the membrane. Okay, so I depolarize right here. So at that point, let me draw in what we're going to have. Okay, so right here, I'm going to have my chemically gated sodium channel right there. Okay, and then sodium is going to come in. But if I don't have any voltage gated sodium channels to boost my sodium levels in here, then what's going to happen? Sodium potassium pumps are going to pump that sodium back out. Okay, so, and because the potassium wants to leak out anyway. Okay, so this is because of the pump. And this is the leak channel. Then it doesn't stay, so let's say that was +4. By the time the sodium would travel all the way down to here, maybe I only have a +1 worth of sodium. Okay, so on dendrites and cell bodies, I have to have a lot of little EPSPs in order to get that neuron to depolarize at the axon hillock. Okay, and then by the same token, then I also have EPSPs, and those are going to happen in a similar manner, except in the opposite direction. Okay, so okay, so there's that. We already got those. Here's an IPSP. Here's an EPSP. So the binding of inhibitory neurotransmitters induces hyperpolarization of the postsynaptic membrane by making permeability to potassium or chloride to increase. The charge of the inner part of the membrane becomes more negative. The postsynaptic neuron is less and less likely to fire. Large depolarizing currents are required. So these would be, let's see, that color won't show up. Okay, so what do we want to remember? Hyperpolarization, potassium or chloride channels, the inner part of the membrane becomes more negative, less likely to fire, and then I need a greater, I should have typed this, greater than threshold would be required if we had been inhibiting. Like if we were asleep and we need to wake up, then we'll need a greater than threshold stimulus to wake us up. Okay. So there's that. So here is, like I was just showing you on the whiteboard, here is what's happening with summation in EPSPs or IPSPs too, for that matter. Okay, so here are two subthreshold stimuli that are far enough apart that they don't summate. So I fire, doesn't get me to minus 55, but I've waited long enough, and then I fire it again. Nope. Okay, so nothing happened. So I sent the signal in a row, twice in a row, but because the stimuli were so far apart, I had no response. But what if I fire them very close together? So this is going to send a signal. Right. Okay, so this one's going to send a signal, and then it's going to send a signal again. Okay, so it just zapped and zapped. It's like you're tapping it in succession. So I depolarized to here, and then, oh, I got another one, and then that took me all the way up to action potential. Okay. Or, so that's going to be temporal summation. This is just like wave summation in a muscle. And we even can call it that if you want. You can call it wave summation, but I always call it temporal summation in neurons and wave summation in muscles, although it's the same principle. Okay, then what if I have two neurons in two different places, but what if they fire subthreshold stimuli at the same time? Boom. So this is like multiple motor unit recruitment. Okay, so I sent two at the same time, and then I went whoop, whoop, and then it gave me an action potential. Okay, and then it went back down again. So this is spatial summation. So, oh, let's just put a little text in here. Increase frequency, whoops, in that one, in wave summation, temporal summation, and in this one, let's do several stimuli at once. Okay, so you can think spatial summation, they were in different spaces. So we could go here. So here's another one here. And just like I've been showing you spatial summation all along, like on the whiteboard. So we stimulated it in a whole bunch of different places with little baby EPSPs, and then boom, I got an action potential. Okay, now I can get spatial summation with EPSPs and IPSPs here where they compete against one another. So this is going to send a signal, this is going to send a signal, and sure, they're going to affect one another. So I sent the inhibitory signal first, so it gave me a depolarizing potential. I mean a hyperpolarizing potential, but then I sent an EPSP at the same time as my IPSP, so it brought it up to here and took it just a little bit above threshold. So it took me away from hyperpolarization, didn't make me depolarize, but it got me more likely to depolarize if I got another EPSP after that. Okay, so just like in wave summation, how do I add up the effects to get what I want? But instead of muscle strength, now I'm doing just creating an action potential. Okay. Now, if we take a look at this, we're going to talk, well, actually, let's do this right now. And then, so we're gonna go back in our notes to a section. So we won't have to do it in lab tomorrow because it's right here. All right, so I showed you, here's what happens when you don't have any voltage gated sodium channels up here at the top, is that you'll apply the stimulus and then the stimulus is going to decay. Or the signal is going to decay. The voltage is going to decay. So it gets less when it gets down here. So maybe it was +4 right here, but now it's only +1 right here, okay? But in non-myelinated axon, okay, so we're gonna talk about what happens with myelinated and non-myelinated axons. Once we get to the axon, okay, that's where we have voltage gated sodium channels. So what happens is that we get a +4, and before it can wear out, I got a +4 again. Okay, and then +4. Well, we should do +15. Let me undo that. Okay, so +15, and then before it has a chance to wear out, oh, I boosted it again, +15, and then I boosted it again, +15, all the way down. Okay, so you can see it's not necessarily going to add up to like a whole ton on the inside because it's still going to try to leak out. But every time I hit voltage gated sodium channels on my axon, then I get a reboost right here to the voltage. So it says because we have these voltage gated sodium channels along the axon, the voltage does not decay. Okay, so just like muscles. However, conduction is slow because it takes time, and slow is a relative term, you guys, because you know, I don't care what kind of axons we're talking about. Things happen so super fast, but some axons carry signals faster than others do. Okay, so in a non-myelinated, oh, you don't even know what myelinated means. We'll go over that in just a minute. So in a regular, we'll just put regular for now, because that's all you know. Okay, so in a regular axon, then it's going to be just like in a muscle cell. But it's slow because it takes time for ions and gates of the channel proteins to move and must occur before the voltage can be regenerated. Okay, so that's what you know to this point. So here's what we're going to do. I'm going to go back to a part of our notes. So I'm going to go way, 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 way back here. We're going to look at a muscle. No, we're not. We're going to look at a neuron. There's the picture that I want. Okay, so we're going to look at actually one more picture. All right, so here's a motor neuron with its axon right here. Now, some neurons that we have in our central nervous system do not have these little bubbles that are around them. Okay, so primarily in our central nervous system, we don't have, well, specifically in the cerebral cortex and in the gray matter of the spinal cord, we don't have these little bubble things. But in lots of places within the central nervous system, and then in almost all of the peripheral nervous system, the axons are insulated. So they're protected with a material that wraps around the outside that's called the myelin sheath. I don't have that word on here, but I have it in another picture. All right, so the myelin sheath is a lipid material that's made from the cell membranes of cells called Schwann cells. Okay, so we have Schwann cell right here, one Schwann cell. So what these Schwann cells do is that they wrap themselves around the axon, thereby protecting the axon from any potential damage that could happen to them. And we'll talk more about the purpose of myelin in lab tomorrow. But they protect the neuron, And in addition to that, they make sure that the ions are not leaking out into the surroundings. So this coating, it's just like a hot dog bun. This would be the hot dog, and this would be the bun, or pigs in the blanket if you wrapped like crescent rolls or something around the hot dog. And so that's going to keep the ions from leaking out and changing the action potential opportunity to happen, I guess. Okay, the membrane potential, I guess. So here's what an axon looks like. So this is the axon, and then this is the myelin sheath, the Schwann cell that's wrapped itself many, many times around the axon. So besides protecting the axon, what else does that myelination do for a neuron? Well, it creates a conduction known as saltatory conduction. And saltatory means to jump. So it says in saltatory conduction of myelinated fibers, the electrical signal jumps from node to node. So we have little spaces on the axon where the Schwann cells don't touch each other. So those are called nodes of Ranvier. And these nodes are chock full of voltage gated sodium channels. So, and we're going to come back to that. Well, let's just look at this right now. Okay, so what is myelin? It's a whitish fatty substance that protects and electrically insulates fibers and increases speed of transmission up to 150 times faster. They're present only in axons, and Schwann cells wrap themselves around the axons many times to form the sheath. And then the neurolemma is just the plasma membrane of the Schwann cell, and it's squished up cytoplasm and nucleus. I'll show you that picture in just a minute. All right, so what are the nodes of Ranvier? Spaces between Schwann cells that allow saltatory conduction to occur. So the impulse travels from node to node down a myelinated axon. Let me just show you what the neurolemma is. It's just simply, once that cell wraps itself around, all of the myelin sheath is all just cell membrane. But what about this stuff out here on the edges? That's all the cytoplasm. That's all been squished out to the edges, and then the cell membrane that's out there as well. Okay, so here's what happens in saltatory conduction. Let me show you this picture. So remember the action potential is going to be generated at the axon hillock. So now the actual depolarization, I'm not going to have any depolarization where there's a Schwann cell because the sodium can't come in. So it's going to jump to here. So I get a depolarization right here, and then all of a sudden I get a depolarization here and here and here and here, because those are the only places where I have the voltage gated channels where the sodium can come in. So this is just like why most animals, when they run, only have like 2 feet on the ground at the same time, and they look more like they're jumping than they're actually running with their feet in touch with the ground. So, you know, if you take a horse, for example, when a horse is running, it gathers all of its feet up, and then it leaps off of the ground and stretches its body out, and then touches down with its front feet. And before it lifts off its front feet, then it touches the back ones, and it pushes off again. And, you know, lions and cheetahs, and I mean, just everything, when they're running fast, that they're actually jumping more than they are actually coming in contact with the ground all the time with their feet. We don't do that. We're always, I mean, we're off the ground a tiny little bit, but really we have a foot on the ground almost every time when we run. We don't hop like kangaroos or bunnies or whatever. But what that does is make it happen much more quickly. So here is where the stimulus is going to be applied, and then the signal doesn't decay, so it's still minus 55 inside of there. And then because we haven't really lost any potassium to the outside leaking out, then when we get to these voltage gated channels, the inside of the membrane is still minus 55. So then we'll just send it again and again and again. So now we're hopping from node to node, which is actually much faster than this form of depolarization right here. So like it says in myelinated axons, conduction is fast. We call it saltatory conduction. Myelin keeps the current in the axons. The voltage doesn't decay much. Action potentials are generated only in the myelin sheath gaps or nodes of Ranvier, and appear to jump rapidly from node to node or gap to gap. Okay. All right. So, so now we have to go clear down here. Okay. So that's that again. I don't know why I stuck it in there. Okay, so then if we want to compare action potentials with EPSPs and IPSPs, you can read, so what is the function of an action potential? Send the nerve impulse all the way down the neuron. What is the function of an EPSP? Short distance signaling. So to just get it to the axon hillock, and then like we said, moves the membrane potential toward threshold. Okay, and then an IPSP, short distance signaling hyperpolarization that spreads to the axon hillock, moves the potential away from threshold generation of an action potential. Okay, what causes an action potential? It's a voltage gated channel, whereas EPSPs and IPSPs are the chemically gated channels. What is the effect of the stimulus? Open sodium gates, then potassium, because remember we have the voltage, sodium opens and then potassium. And now we're just opening the chemically gated one right here. Remember, low sodium in, but a little bit of potassium goes out too, but remember it's mostly sodium. And then I said that IPSPs are either potassium or chloride channels or both, okay? And then how do they repolarize? Well, the gates just close once we get back to resting membrane potential, well, once we get below -55. This just fades away, okay? So I don't have to do any repolarization. It just, the membrane changes with time and distance. Shows how far, positive feedback cycle, yeah, one gate opens the other and another not happening in these. And then remember we said +30, 40, 50, that's close. Remember, this is going to be hyperpolarized, so it's going to go towards -90. And then no summation, because remember, once you get an action potential, it travels all the way down. So it doesn't get stronger and stronger and stronger as it goes, but we absolutely have summation. So like we said, graded potentials, depolarizations, baby depolarizations, baby hyperpolarizations. Absolutely, we have an absolute and relative refractory period and action potential, but we don't have them in the EPSPs and IPSPs. All right, so just to finish up our part on temporal summation and spatial summation. So temporal summation, again, occurs when one or more, and usually just one, but here's the important thing, is that we want to remember that it's a rate increase. Okay, so one or more presynaptic neurons transmit impulses in a rapid fire order, so rate increase. We want to send the signal faster and faster and faster. Okay, so bursts of neurotransmitter are released in quick succession. So one EPSP comes right after the other and they summate at the postsynaptic neuron. Okay, and so that's one, and we can do the same thing with IPSPs. Make it harder and harder and harder for that neuron to receive the signal. Okay, so just send it super, super fast, one right after the other. Spatial summation, on the other hand, is going to be still the same, still summation, but it's stimulated at the same time by a large number of axon terminals from, can be the same, but usually different neurons, okay? And so many EPSPs or IPSPs occur at once. Okay, so let's see. Looking at our schedule, I'm kind of ahead of schedule. When is our test? Oh, our test is Friday. Oh yeah, no, we're great. We're perfect. Okay, so I am gonna do this last little bit, and then Wednesday, we will have an opportunity to do the Kahoot and watch a little thing on neurotransmitters. Okay, no, we are perfectly fine time-wise. Okay, so let's talk about neurotransmitters now to finish off this stuff. Okay, so what are neurotransmitters? Well, we already know. They're the means by which each neuron communicates with others, because we're going to cross the gap, cross the synapse, because we have no other way of crossing the synapse, okay, for the signal to get from one to the other, because they don't touch each other. All right, so here are some different groups of neurotransmitters that I want you to know what they do. Okay, so the first one, acetylcholine. You're totally familiar with how acetylcholine works, okay? It's excitatory in skeletal muscles. Sometimes it's going to be inhibited by the botulism toxin. So remember we talked about tetanus. So the tetanus toxin, when you step on a rusty nail and the organism grows inside of you and releases its toxin, it goes to the neuromuscular junction. When it does that, it binds like it's acetylcholine to get the neuron to fire. But in botulism, which is made by a cousin to the tetanus organism, that botulism is going to block the release, inhibit the release of acetylcholine, so that you can't get muscle contraction and you have paralysis. So botulism is the kind of food poisoning. If you ever see like a puffy can, don't open it. If you push on the, like if the lid is, the top is bowed out, or if you push on the lid and it clicks, throw that can away, because that means we've got some anaerobe growing inside of there, produced a gas, and it's going to kill you if you eat it. So don't eat it. Okay, binding inhibited by curare. So curare is a plant toxin that native peoples in tropical areas have collected this toxin, put it on the end of their blow darts, and then so when they go out hunting and they blow the dart out and it gets into, you know, like a capybara or, you know, some other kind of animal that's going to run away that lives in the tropics, as soon as that curare gets into the bloodstream, it's going to go to the neuromuscular junction and prevents the acetylcholine from binding. So the animal is immediately paralyzed, so it just falls down, and then the hunters just run after it and pick it up and butcher it, you know, or whatever they're going to do with it. But they can be quite a distance away and shoot their blow darts. As long as that curare gets into their bloodstream, then they're going to be paralyzed. Snake venom also works like that, neurotoxin. So when a viper, so you're on rattlesnake just bites its prey and then lets go, and then waits for the little mouse to fall down, and then it goes and eats it. It doesn't have to hang on to it like a constrictor does, because when a constrictor eats its prey, then it bites it and holds onto it and then wraps its body around the prey. But poisonous snakes don't have to do that. They just bite it and then wait for the snake venom to inhibit it, and then they go find the thing they just ate. I mean, they just bit, and eat it. And then we have some problems with acetylcholine transmission. We may get decreased acetylcholine in Alzheimer's disease, or receptors get destroyed in an autoimmune disease called myasthenia gravis, so that muscle contraction and weakness occurs on muscle contraction doesn't happen, but it's a autoimmune disease. And that's just what acetylcholine, you can read that if you want. Norepinephrine is our excitatory neurotransmitter during sympathetic stimulation. And oh, I should put that in there. Let's see. So let's put this. Okay. So it's the neurotransmitter of sympathetic stimulation. So it's a cousin to epinephrine or adrenaline. We used to call this noradrenaline. And so it's all the stimulatory effects that we have when you want to run away from a Rottweiler, then that's going to be because of norepinephrine. Why do people do meth? Because it causes more norepinephrine, and we'll take a look at that on Wednesday. More norepinephrine to be released. Okay, so that's why people do uppers. And then cocaine blocks its reuptake, so it stays in the it stays in the synapse longer. What do I need you to know about dopamine? So dopamine is also, it's going to act like cocaine. Well, it's going to stay in the gap like norepinephrine will because of cocaine. It is the neurotransmitter that makes sure you don't have Parkinson's disease. So when you have Parkinson's disease, you don't have enough dopamine. We'll talk more about that on Wednesday. May have it in schizophrenia. But here's the important thing. It's our reward pathway neurotransmitter. And so when there are substances that increase our release of dopamine, we can become addicted to those. And heroin is a really good example. So again, we'll talk about that on Wednesday. Okay, then we have serotonin. Serotonin is an inhibitory neurotransmitter. LSD, you don't know what that is. Well, you might know that, but that was a drug from a really long time ago. We'll talk more about tomorrow. It helps with sleep, appetite, makes you feel good. It is part of our feel-good neurotransmitters, especially with clinical depression. So Prozac and other, they're called SSRI medications that are called selective serotonin reuptake inhibitors. So what that means is if I am clinically depressed, then this is going to, if I take Prozac or another SSRI, it's going to make sure that the serotonin stays in the gap longer so that I can get those effects of feeling good and not be depressed. I like serotonin when I eat. I get real happy when I eat foods that have tryptophan in them, because like turkey does, because tryptophan is what serotonin's made out of. And so when you feel really good after a meal, then you got a lot of serotonin release. So one time we were at this restaurant we used to like to eat where my in-laws used to live, and so I just finished eating, and I said, oh, my serotonin levels are off the charts. And my son, he was like 12 or 13, something like that, turns to me and he goes, Mom, you always say things like that, like my coccyx is twisted in a vertebral fashion. And I'm like, you're the weirdest kid ever. And so anyway, he just had to give me a bad time about how I say weird things myself, and he made up some weird thing that's not even, doesn't even happen. All right, GABA. GABA is a really important inhibitory neurotransmitter. So we're going to look at, well, no, we're not. Okay, so why do we need GABA? Well, remember I said if we're going to flex our elbow, we need the biceps brachii to contract, but I need to inhibit the motor neurons for the triceps brachii. So up in my brain, I'm going to release, or in my spinal cord, I'm going to release GABA to inhibit the motor neurons that I don't want to fire. So as I'm walking down the street, so that my muscles can act in opposition to one another, the ones that I want to contract, I'm sending acetylcholine to, and the muscles, the neurons that I don't want to fire, I'm sending GABA to. And so I just trade off ACH GABA, so that I can walk, just merely to walk across the street. I've got all that neurotransmitter secretion going on. Now, we'll also get more GABA release with alcohol, and we'll look at that on Wednesday as well. So you know how alcohol affects your decision-making process and your response time, and it's a general inhibitor of your central nervous system because it causes GABA to be released. And so we can't react, we can't do things as well under the influence of alcohol because there's too much GABA. Glutamate is a general excitatory neurotransmitter. We use it in learning and memory. The interesting thing is when we have a stroke, when our neurons are starved of oxygen, we release tremendous amounts of glutamate that could actually stimulate neurons to death. Usually that excess glutamate release would be caused because of a stroke, blocked blood vessel. So anyway, but it is one of the major excitatory neurotransmitters in the brain, not like acetylcholine, because that's on our muscles. And then you can read about other things too. Now, endorphins are fabulous because they're inhibitory. They are the ones that are going to inhibit substance P, which is the neurotransmitter that causes pain. And it's mimicked by morphine because it's actually made out of the same class and stuff, and so it's going to bind to the same receptors. Heroin is also an opiate in the same family, and heroin and methadone. So and Noxolone. So why do we give these? Well, because we want to stop pain. So that's why a lot of times people, when they take morphine, we're going to find out how do they become addicted to that, like in heroin. And but that's why we want those endorphins, but our body makes them ourselves. So you know how sometimes you may be really involved in something and then you look down and all of a sudden you're like bleeding and you're like, well, how did that happen? And then it starts to hurt. Well, that's because endorphins were released so that it can help you through that, you know, that painful time to save you. But here's another good example. If you've ever heard about someone who has a runner's high, where they just run till they're just dying, and then all of a sudden you feel really good, you know, like you hit the wall and then you got pushed past that, and then you're like, oh, I love this feeling, I'm feeling so good. It's because your body is dying from pain, but your brain goes, nope, we're going to keep going, and releases endorphins so that you can. So that's what people who have a runner's high are looking for, is that endorphin release. And then lastly, substance P is the one that gets our pain stimulus. Okay. All right, so that's all of our notes. So we've been through everything. So our test on Friday, let me stop sharing. So our test on Friday will cover all of the muscle physiology stuff, all of our nervous system notes. Tomorrow in lab, we'll do some more to make sure that we understand what's going on. I'm going to show, no, I won't show you that next week in lab. No, I'm not going to show you anything yet. But yeah, so we'll just, and I don't think lab's going to be very long, but it'll give us the opportunity to talk more about, you know, some other things. Well, maybe I'll put the, okay, so here's what we're going to do. Sorry that I have to think through things in real time. So I'm going to show you the little video thing on neurotransmitters tomorrow in lab, along with what we were going to do in lab, structure of the neuron. And then Wednesday will just be review. So we should be able to Kahoot. We tried it on Friday with my 202 class because they had a test today, and it worked real good. So you can Kahoot from home just as well as you can Kahoot, you know, when we're doing it here. So we'll be Kahooting and reviewing on Wednesday, and then Friday, you'll need to take your exam by Friday, but we'll still have class meeting, but we're going to go on to new stuff. We're going to start talking about the cerebrum on Friday. So I'll open up the test. You can probably, I'll open up for for Thursday and Friday. So you absolutely have to have the test done by Friday, April the 10th at 11:59 p.m. Boom, or it's going to cut you off if you don't get in at least an hour before that. You'll have the hour to do it like we normally have lecture class, but you'll just do it in Canvas. Okay, so I think that's all. And so come back tomorrow at 1 o'clock and we will have our regularly scheduled lab class. So appreciate you guys that came and hope that you got what you needed, and we'll stop the recording and we'll see you tomorrow. Okay, bye.